Welcome to the next panel on therapeutic approaches for skeletal muscle. It's a pleasure for me to introduce this panel that is focused on skeletal muscle, which is a fundamental tissue for voluntary movement and metabolism, and defects in its function severely affect quality of life of several types of patients affected by many diseases, and in case of degenerative diseases, also significantly shorter lifespan. So there is a strong interest in trying to define therapeutic strategies to be able to extend or improve skeletal muscle function. Skeletal muscle is maintained by endogenous muscle stem cells that are required, are essential for postnatal muscle growth, tissue repair, and also skeletal muscle homeostasis. So this cell type, of course, are the primary interest as a tool or a target for developing approaches and therapeutics to promote tissue repair and extend skeletal muscle function. In thinking about this panel, we decided to focus on three main areas regarding skeletal muscle research. So the first speaker, Andrew Brack, associate professor from UCSF, he'll focus on skeletal muscle aging, in particular the effect of aging on the muscle stem cell compartment, and also how they respond to specific stresses, and how that affects also the heterogeneity of the stem cell compartment, and what are the implications of these changes in muscle stem cell function. And the second speaker is Rita Berlingueiro, professor from University of Minnesota, and she pioneered studies in the derivation of myogenic progenitors from human ES and iPS cells, and by expression of defined transcription factors. And she will tell us about today the status of her studies on this approach, which of course has several implications because it generates tools for drug screening, gene correction ex vivo, but also with the hope of generating large number of progenitors that might reach a clinical relevance when actually transplanted into patients. And then the third speaker is Pier Lorenzo Puri, professor from Sanford, Burnham, Prebys here in San Diego, and he will focus on the interaction of muscle stem cells with the tissue microenvironment, specifically which signal muscle stem cells are exposed to in healthy and chronic conditions, and how these signals are actually received by muscle stem cells and translated into epigenetic changes. And he has done exceptional work in this area, and he led recently a clinical trial with boys affected by Duchenne muscular dystrophy with givinostat, which is an epigenetic drug. This is just the introduction, so it's a pleasure for me now to introduce the first speaker, Andrew Brack, associate professor from the Department of Orthopedic Surgery at UCSF. Welcome.